I just want to introduce Dr. Harshita Tiwari, ma'am, and she has very good experience. She done her masters from MS from Naipur, Kolkata, and PhD from CSI Triple M Jammu. She also worked in Solaris Therapeutics, and right now she is working in a drug chemistry research center as a computational research scientist. Today's lecture on network pharmacology. So, network pharmacology is an interdisciplinary field that integrates bioinformatics, systems biology, pharmacology, and network science to study the interactions between drugs, proteins, genes, disease, and biological pathways within a network framework. Today's lecture is going to take by Dr. Harshita ma'am, and hope it will be enjoyable and experience. You will get some experience about the network pharmacology. Over to you, ma'am.
Okay, so hello, a very good evening to all of you. My name is Dr. Harshita Tiwari. I am working as a computational research scientist with Drug Chemistry Research Center. So, in today's lecture, we will provide an overview about the network pharmacology, and we will how it is important for all of us, so that you can get a idea related to the field of network pharmacology. So, here I am beginning. First, let's get started with the definition of network pharmacology. So the network. So the network is generally consisted of two or more things that are linked in order to share a resource. So network can be anything like uh, a social network in which two or more than two people interact with each other. Okay, so that they can say share the resources or information. It is a kind of network. So, like this, we have a biological network inside our body as well. So, and the pharmacology is the study of how drugs and chemical impact living organisms. Okay, so these two terms, that is network and the pharmacology, these two together makes the field of network pharmacology. So, by definition, network pharmacology is a discipline that integrates system biology, bioinformatics, pharmacology to understand the complex interactions between biological system and drugs at a molecular level. So we all know that our body it is not consisted of one molecule. Okay, so our body is very complex system which consists of several networks which are continuously talking with each other. So we have biomolecules like proteins which can interact with each other as well as other biomolecules like RNA, DNA. So we have regulatory gene networks in which the regulatory units of our body, they interact with each other and regulate a particular biological process. Okay, so they can provide the signals to so that a biological process can be carried forward or can be halted. Likewise, there are regulatory networks and the metabolic networks as well in our body. So this network is very important for the proper functioning of the biological system and to maintain the homeostasis of a particular bodily function. Okay, so by the application of network pharmacology, we study the interaction between these different biomolecules, and we can conclude which interactions are important and for a particular mode of action to take place inside the body. There are various applications of network pharmacology in today's medical fields. Like, as we know that the globally there are different type of traditional medicinal systems. So these traditional systems are being practiced by the folks of a different regions of different countries. So these traditional systems include the use of different herbs and the minerals so that for the cure of a particular disease or the ailments. Okay, so like in India there are different kind of traditional system of medicine like Ayurveda, Siddha, Unani, and there are African medicinal system, traditional Chinese medicinal systems. So these are basically dependent on the application of natural sources, and we have heard from the mouth or the text text that these are very important to cure a particular disease, but we don't have any kind of scientific evidence to support this, to support that to support how these traditional system of medicine is working and how it is curing a particular disease. Okay, so this lack of scientific evidence false or create a sense of doubt in the peoples or the scientific community. Okay, so in this context, the network pharmacology comes as a very good tool to understand the mechanism, or to provide a particular scientific evidence behind this traditional system of the medicine. Okay, then it as we know that we use different kind of formulation in a traditional system of medicine. Okay, we use syrups, we use asavas and decoctions in traditional system of medicine, but we don't know how these system of medicine, how these different formulations or the herbs are interacting in that particular formulation. Okay, so we can understand that too with the help of network pharmacology. Okay, then it we by studying different kind of plants, or by studying their different kind of the phytoconstituent of that particular formulation, we can design a new formulation based on the plants. Okay, then the botanical biomarkers for good quality, so we can study the network and the formulation of the composition of the plant with the help of this network pharmacology, and we can describe the botanical biomarkers as well with the help of network pharmacology. Okay, then with respect to the pharmacology, so it has to develop new leads from the natural products. So we know that the plants are a complex of different phytoconstituents, and in the traditional system of medicine, we basically uh, goes for a holistic approach in which we don't isolate a particular compound, rather we take a complete formulation or a complete plant as an or a particular complete part of a plant to as in medicine. Okay, so by the help of this network pharmacology, we can identify a particular constituents which is important for a particular activity. Okay, so then after identifying these particular lead compounds, we can go for the we can develop that we can study that particular phytoconstituent in a detail, and we can use, if if a desired effect and there is a balance between the side effect and the effect of the plant, means that if we get the desired activity, we can take that particular compounds as a lead compound. Okay, so lead compound basically, whenever we study the medicine or the drug discovery, we describe uh, the compounds in terms of hit, lead, and Cs and the drugs. Okay, so generally the primary compound which have a biological activity, but we that might not have a particular or desirable peak, pharmacokinetics or dynamic profile. Okay, so that primary compounds from the first screening is known as the hit compound. Okay, then the lead, when we optimize the PKPD or the side effect and the effect of that particular hit compound, then that compound is known as the lead compounds. Okay, so it can help us to find those lead compound as well. Then understanding the mechanism of action of the drugs. So a particular if we have a particular compound in our mind, okay, or the drug, and we don't know how it is interacting in biological system, so we can take the help of network pharmacology to understand the mode of action of that particular drug compound. Then they determine the possible side effect of the drug. As I told that our body is a complex of the different processes. Okay, so study we can study the network of that disease, and then we can understand how these molecules are interacting with the different molecules and causing the effect as well as the side effects. Then predicting new indications. So new indication is basically represent the term, it is a term used for the drug repurposing. Okay, or drug repurposing. So we can study the compound, and we can find the new indication or the new diseases in which it can be used. then the possible drug and drug interaction, so we can compare the network of two different drugs. Okay, then we can find the possible additive synergistic or the contrasting effect of two drugs. Then the drug repurposing, it is similar to the new indications. Okay, then drug research, the third field is drug research. So it helps to identify novel drug targets. Suppose we have taken two compounds and we are studying their biological networks. Okay, then we know that it act on this target, and it acts on this target by a particular route. Okay, but when we make a particular biological network, so if we want to inhibit or to modulate a particular target, so there can be different paths which can lead to a particular target. Okay, so there can be various processes which can activate or halt a particular process. So these paths can be identified with the help of network pharmacology, then reduce cost and time in the in silico evaluations. So I told you that we can understand a particular mode of action, or to understand various aspect of a particular or new drug entity. So by these we can reduce the cost of drug development. Then understanding the signaling pathway of disease type, so we can compare the the pathway of of a diseased individual with the non-diseased individual. Okay, so by comparing the different networks, we can get an idea what is a basic important pathways which are involved in the disease progression. Then the therapeutics for multi-gene dependence disease, so there are some diseases like cancer, Alzheimer's, which or diabetes, which doesn't involve a particular pathway. So basically these are multifactorial diseases. So there can be different factors or the multiple factors which are important for the disease progression. Okay, so these multi-gene or multifactorial diseases can be easily understood with the help of network pharmacology. Then discovering of the disease causing genes. So by studying a particular pathway, we can find out the genes which are important for the disease. Then diagnostic biomarkers. So as I told you that we can compare the genetic or the biomarker profile or the profile of diseased and the non-diseased individuals. So there will be some genes or the proteins which are more expressed in particular disease conditions. So these identified genes or the proteins can serve as a biomarker of in a particular disease. So we can identify this with the help of network pharmacology. Then studying drug resistance and the antibiotic resistance. So similarly, when we compare or build a network of a particular drug with the disease, like microbial diseases, so we can easily identify which genes are responsible for drug resistance or the antibiotic resistance. Okay, so the data basically this can be done with the help of, we can generate the genomic profile or the proteomic profile of a particular cell after treatment of a particular antibiotics, which then we can generate a network of a network of that particular treated cell. Then we can identify this drug resistance and the antibiotic resistance gene. Then moving towards the workflow of the network pharmacology. Okay, so I told you the application of network pharmacology. So now we will discuss how we can attain or to study the different we can perform different tasks with the help of network pharmacology. So for the network to perform a particular network pharmacology task, first and the most important task is to acquisition of the data. Okay, so data mining is the initial step involving the identification of the active compounds and the disease related genes. often through literature searches or the public databases. Okay, so active compounds we will say when we are dealing or we are dealing with a particular plant. Okay, so plant are the composed of several phytoconstituents. So we need to suppose I have provided a particular plant that is a Vidania somnifera. So it might consisted of different or the several active compounds. Okay. So our first aim is to make a list of all the reported compound of that particular plant. uh, these there are different databases which can help us in this task. Okay, then the disease related targets. So likewise when we are talking about a particular disease, suppose diabetes. So there will be a there can be means publicly available data for that particular disease, so we can collect those data, and that can serve as an initial point for the network creation. Then once we have collected all the data related to the active constituents and the drugs and the protein interactions which are involved in a particular disease conditions or the drug targets or the phytoconstituent targets, then the second aim of our work is to construct a network with the help of that particular data. Okay, so the network construction and visualization aims to provide aims to predict disease related genes, identify common genes between disease and compounds, and understand the mechanism of the medicinal plant in disease treatment. Okay, so first we will we with the help of collected data, we will construct a raw network of all the interactions we have attained from the data mining. Then once we have created a raw network, then the next step will be to analyze that network. Okay, so the network analysis via intuitive visualization of complex biological networks, aiding the interpretation of the network structures and the relationship, thereby facilitating the identification of potential targets for the validation. That is, it means that after we have constructed a network, there are several approaches or the techniques by which we can identify which interactions are important for a particular function. Okay, so that comes under the network analysis. Then once we have identified important target from the network analysis, we will go for the validation of our result of network pharmacology. So this can be done with the help of in vitro, in vivo, or the in silico experimentation. So majorly it can be done with the help of molecular docking, gene expression microarray analysis, RT-PCR, Western blotting. So these are different the techniques which can be used to verify our results. And once we have performed all these tasks, we can go for the publication or to report the study. Now, I will talk more about the data mining. Okay, like what kind of data we need for a particular task. So suppose I want to work with the different drug molecules. Okay, so these I have categorized under the category of the compounds. So there are various publicly available data sets like PubChem, Cambial Drug Bank, which contains the information related to the structure of those compounds, their predicted targets, okay, and the related bioactivities. So these compound data sets serves as a primary point to collect the information related to different compounds. Okay, then there are particular targets. There are particular databases which deals with the natural compounds only. So like Dictionary of Natural Products, Impact, Dr. Duke's Phyto database. So Dictionary of Natural Product, it is an paid or commercial database. Some of the functionality of this Dictionary of Natural Product data sets are freely available. Okay, then Impact, this data set is basically for the Indian medicinal plant. It is freely available for to use. Then Dr. Duke's, it is also a freely available data set of natural product. So these are few names. There can be more data sets for this task. Okay, so this data set, when we want to deal with a particular plants, so then we want to look for the data set which deals particularly with the natural products. In that case, we can get the information such as the phytoconstituent present in a particular plant, their chemical structure, from these natural product databases. Then, once we have information related to compounds on the natural products, there are certain data sets by which we can get the information related to the interactions of these compounds, phytoconstituent or a chemical compounds, like what are their targets, and how these targets are interacting with each other. Okay, so there can be different databases. Some three data sets I have written here, like stitch, string, distinct. So stitch and string, these can provide the information related to the protein or the gene, protein gene interaction. Then distinct, this is particularly related to the diseased condition, in which you can get the information related to the interactions in a particular disease conditions. So apart from that, there can be multiple data sets like biocata, which can be used for this particular task. Okay, then these interaction data set, there can be other other data set like compound target data sets, like drug bank, therapeutic target data set, TTD, cc server. So TTD and drug bank, so it provides an authenticated route for a particular compound target acquisition. The c similarity server, it is server in by which we can predict the targets of a particular compounds. Okay, suppose we have a new chemical entity or a new chemical compounds whose target information is not available. So what we will do, we will go to the c similarity server, we will provide the structure of that particular compound. Then we can get the information related to, means these interactions are the predicted by the c server, which can be probable interaction of that particular compounds. So this server is very useful when we want to predict the target of a particular compounds. Okay, so these are basically the important data sets for the data mining. Then once we have collected these primary data, we go for the network construction and the visualization. So this is a comprehensive view of a network. So network is basically composed of the node, edge, hub, and the module. So node, it is a fundamental unit representing a biological entity of component within the network. Okay, so it can be a protein, a gene, okay, a metabolite. Okay, so this is these different units which are presented or represented as circles. These are different nodes in a biological network. Then the edge, so edge is basically the connection which connects two different nodes. Okay, so a connection representing a relationship or interaction between two nodes in the network is known as the edge. Then the hub. So hub, a highly connected node that plays a significant role in network communication and the function. So you can see here some nodes in the biological networks are connecting, or the major points by which multiple nodes get connected. Okay, so these kind of nodes which are highly connected from the other nodes are known as hub. Then the module. So in a biological networks, there can be different sub networks. Okay, so it is a cohesive group of nodes within the networks that are densely interconnected and often represent a particular function unit or the pathway. So when we identified that with different methodologies, that this part of this particular network is important for a particular biological function. Suppose from this network we have identified set of proteins which are involved in the AKT pathway or the cancer pathway. Okay, so we will say that this is a module which is responsible for the anti-cancer activity. Okay, so these functional units are known as the modules. So I hope it will be clear what is node. So node, basically a particular entity. Then these nodes are connected by the edge. Then the hubs are the nodes which are highly interconnected. Then when we get a particular set of genes or the PPIs which are responsible for a particular task, then it is known as the module. Then the network analysis. Okay, when we once we have formed a particular network of a gene, then we analyze those network with the help of different network analysis approaches. So one approach is the clustering analysis, then the topology analysis, then the pathway analysis, then the functional enrichment analysis. So clustering is there are different kind of clustering algorithms which we can apply on this the primary biological network we have created. Then we find the genes which are highly connected, or the genes which are densely connected with each other, or which can be perform a particular task. Okay, so this identification or the grouping of different interactions is known as the clustering analysis. Then the topology analysis. So topology analysis, it involves the examination of the structural properties or patterns of the connections within the network to understand its organization, dynamics, and the functionality. So topology analysis is basically when we try to understand how different nodes are connecting with each other, like how many nodes are connected with different nodes. What is the centrality of that node? It is known as a topology analysis. Then the pathway analysis. Pathway analysis is that when we identify or interpret a particular biological pathway, okay, from a particular biological network, then it is known as the pathway analysis. So suppose we have a particular information in the literature that these set of genes are responsible for a certain pathway. Okay, so we try to identify these interconnected genes form a particular biological network. So this is known as the pathway analysis. Then the functional enrichment analysis. So functional enrichment analysis identifies biological functions, pathway, or processes overrepresented in a gene or protein set to gain the insight into their functional significance. Okay, so this is basically an enrichment analysis when we try to identify or to justify that these are really important for a particular task. So these different tasks can be performed with by using different measures like clustering analysis. So there can be different types of clustering algorithms like k-mean, modularity, density. Okay, so these different algorithms can be used for the clustering analysis. Then there are different modules to perform the topology analysis, like what is the degree, means how many nodes are connected, then the clustering coefficient. So these are path length analysis. Like suppose they as I told you that a particular task can be performed by using different pathway in biological systems. So what is the shortest path by which a task can be attained? Okay, so this can be used by the path length analysis. Then the pathway analysis. So we annotate a particular set of genes or the protein interactions to find out the best possible pathway from the network. Then the functional enrichment analysis. So there are different pathways which are biologically validated, like in and the information related to their path their function is present in various databases such as KEGG, GEO databases. Okay, so these databases can be used for the functional enrichment analysis of a particular set of genes or the pathway in the network analysis. So after performing all these analysis, we are able to identify the major player in a particular networks which can play a significant role in a particular biological function. Okay, so once we have identified a set of targets or the node hub genes from this network analysis, we can perform this task with the help of one tool which is known as the cytoscape. Okay, so cytoscape, we can visualize our network. We can perform different type of tasks like topology analysis, clustering analysis, gene enrichment analysis. Okay, and this is a freely available data softwares, so you can install this cytoscape in your system and perform these different tasks. Okay, then once we have performed the network analysis of our of different biological process, we can validate our results in with the help of in vivo experiments as well as the in vitro experimentation. Okay, so this was all about the basics of the network pharmacology, like how we can build and validate our finding. Then let's start by taking one case study. Okay, we will learn how this is involved in the drug repurposing. Okay, so drug repurposing involves identifying new therapeutic uses for existing drugs beyond their original intended interactions. So classical view, we generally sees that we say that a particular drug exert a particular action by acting on a particular target. Okay, then we also see that whenever we take a particular drug, there can be side effect of that particular drugs. Okay, so these are known as the off target effects, that can be adverse effect or only side effects. Okay, so that was the classical view of the drug action. Then if we will see it as a systems view, like by taking the whole body as a system, we will see that if we are taking a particular drug, it is exerting effect on it is affecting a particular pathway or the target, which can modulate a particular system and exerting a effect or the desired effect as which is indicated for that particular drugs, while this it might act on the other target, or it can also by interacting in the indicated pathway, it is altering the effect of a particular node, which can lead to the adverse effect. Okay, so this is the systemic view in which we can say that a drug can act on a particular compound can act in two or multiple targets and exert different kind of effects. Okay, so when we study the drug repurposing, what we study, we there is a drug which, suppose a paracetamol, which has a particular action, and we will say that it is its primary target or the primary indication. But at the same time, we will say that it can act on other targets in our body as well, and it has the capability to affect the other systems in our body. Okay, it has the capability to affect other systems in our body too. So these other systems can play the major role in some other diseases as well. Okay, so by altering or by studying those side effects, we can find the the different other diseases in which paracetamol can act. So this kind of findings in which we find the other indication or other uses of a particular drug is known as the drug repurposing. Okay, so uh, uh, let's more understand it more by taking the example of the thalidomide. Okay, so as we all know that this is a very infamous drugs. So this thalidomide was first marketed in 1957 in West Germany. So it was basically given to the pregnant ladies which have anxiety, trouble sleeping, and with the morning sickness to ease these conditions. So what people saw that the pregnant women which take this thalidomide are giving birth to the babies which have seal-like limbs. Okay, so this condition is known as a phocomelia. So due to this phocomelia or the teratogenic effect of this drug, the drugs was withdrawn for the market. Okay, but now people have studies that these drugs can exert the other effect as well, like it is basically an immunomodulator. So it is now used for the treatment of multiple myeloma, tuberculosis, leprosy, and graft versus host diseases. Okay, so as to simplify this, I will say that the primary indication of this drug was for the to ease the stress in the pregnant ladies. Okay, but which leads to the teratogenetic effect, and drug got withdrawn from the market. While when we study that the thalidomide in detail, after some time, we find that it has capability to interact with the other system of our body as well, and these interactions can be very useful to treat other diseases like multiple melanoma and tuberculosis. Okay, so then this drug which was previously withdrawn for the market is repurposed for these other disease conditions. Okay, so this is the case, particular case of the uh, for the drug repurposing. So now this drug, thalidomide, is also studied for the treatment of the ulcerative colitis in a recent study. Okay, so and this study in which the treatment of the ulcerative colitis was done with the help of thalidomide is experimentally validated too. Okay, so what they did, they simply take the thalidomide and ulcerative colitis. Then, with the help of the datasets such as PubChem, PharmaMapper, they have identified 133 targets which can be potential target for thalidomide. Okay. Then, from the gene cards, PharmaKB and the drug bank, they have identified 2449 targets which are involved in the ulcerative colitis. Then after that, they found that the 121 targets which were the common targets of the thalidomide and the ulcerative colitis. Then by taking these 121 target, they what they did, they perform a protein network of that 121 targets. Okay, so after this they have identified the hub targets like src, egfrj, jack, stat, mapk, rac1. Okay, so these are basically involved in the inflammatory processes of the ulcerative colitis, and these they checked these identified identified hub genes with the protein networks. Then what they did, they perform a molecular docking of these identified targets with the thalidomide. Okay, then after molecular docking, they find that after molecular docking, they perform the gene enrichment analysis and the hub target expression with the help of Go, KEGG, and the GEO data sets. And after these analysis, like target prediction, PPI, molecular docking, and the Go and the GEO analysis, these are all in silico analysis. They perform the in vitro experimentation of these targets of the identified target to see the expression of these identified hub genes in the presence of the thalidomide. Okay, so what they identified, they identified the expression of certain genes. Okay, the expression of certain genes like MAPK and JAK2, RAC, STAT. It is significantly affected with by using the thalidomide. Okay, so they find that they did the go enrichment analysis, and they find that certain kind of genes which are involved in the LPS induced ulcerative colitis and increased by in these disease conditions are basically decreased. The expression of those genes are decreased in the when we treated the ulcerative colitis condition with the help of thalidomide. So as you can see in left hand side, there is an LPS induced pathway, then the treatment done with the thalidomides. So the genes basically the PI3K, PI3K, AKT. So which was and the EGFR pathway. Okay, the JAK pathways, which are basically different in case of the LPS induced inflammation. They are now they are now decreased or modulated in case of the thalidomide treatments. So so this was a case in which we studied the effect of the thalidomide um, in the ulcerative colitis condition, and then we find that they can it can be used in the ulcerative colitis. And the study also provided the evidences based on the wet lab studies that related to which genes are affected by the treat by with the treatment with the thalidomide. Okay, so this was one case in which we repurpose a particular drugs for a particular conditions. Likewise, there are several other studies ranging from the and cancer to the Alzheimer disease, which have used this network pharmacology approach for the identification of disease targets as well as for the new indications. Okay, then the another case is the study of the or to deconvolute the mechanism action of the traditional medicine. Okay, so as we know that the plants, they are collection or the cocktail of different kind of phytoconstituents. Like the phytoconstituents which are present in a plant, they can be belong to categories like polyphenols, pigments, sterols, terpenes, others, and the polysaccharides. Okay, so all these so if we will study a particular plant with respect to a particular disease, so certain kind of chemicals will be important for the treatment of a particular disease, while certain can be important for the treatment of other kind of disease, right? So we need to study which phytoconstituents are important for a particular disease condition, and this can be studied with the help of network pharmacology. So the major steps which when we study a particular plant or the formulation with the network pharmacology involves first approach is to identify the medicinal plant. Okay, then the data mining, as I told you. So data mining can be done with the previous publicly available data sets, literature, or the lab experiments. Okay, so you can you can perform the database searches or the literature searches, or you can simply extract the natural compounds of your plant and make a data set of like these compounds are present in this particular plant. Okay, so once you have identified these plants, you can search for the target of that particular plant in the reported databases, or you can go for the C searches, C search similarity server to find or to predict the target of that particular phytoconstituent. Okay, so now you have the targets which are for the plants. Okay, then on the second part is to identify the targets of the disease. So you will check which are the common target of that disease and the plant's phytoconstituents. Okay, then once you have identified those target, you will try to perform to perform the network pharmacological approaches. Like you can go for the pathway analysis or to gene enrichment analysis. Okay, to identify those particular targets or the modules of that particular biological networks. Okay, so once you have validated, you identified a particular the particular targets which can be important for a particular disease, you can go for the validation in silico, in vitro, or the in vivo. Okay, so this is basically for the plant. One thing important here is to go for more customized data sets for the plants and to predict the targets of the particular of that particular phytoconstituents. Okay, because for most of the plant constituents the targets are not available. Okay, so you can go for the similarity based target prediction. And the other thing is that to in case of the drug repurposing, basically we are using the drugs which are already approved. So their pharmacokinetics and the dynamics property are basically acceptable. While in case of the plants, mostly what happens that these phytoconstituent violates the principles like Lipinski's rule. They violate the principles. So we need to find out the phytoconstituents which have drug-like properties. So this is the basic difference when we perform the drug repurposing and the plant-based network pharmacology. Okay, so these filters you can always apply. So after applying all these filters, once you have identified the particular targets, you can validate those targets in the lab, and then you can report the study. Then, whenever you are conceptualizing the network pharmacology in plant research, the first aim to understand the complexity. Okay, so that recognize that plant contains numerous bioactive compounds with diverse molecular targets, requiring a system level approach for the analysis. Okay, so we have to make make up our mind that whenever we are dealing with a plant, it might have the scaffolds or the chemical compounds which are very complex in nature. Okay, so this is the first step. Then the network representation. Envision plant as a complex networks where bioactive compounds interact with the target proteins, forming a network of the interactions. Then we will we will represent in our mind that how these plants can be talk with each other as well as with the biological system. Then the integration of the data. Then incorporate data on the compound structure, target proteins, biological pathways, and the interaction into the networks. So these data you can find from the different various literature sources or the databases. Then the functional context. Then it is very important to validate our findings for go for the gene enrichment analysis. Okay, and then you can also integrate the multi-omics integrations. So if you have good facilities or you have the data which are coming from the genomics, proteomics, metabolomics or the proteomics, like you have the chances to get the omics data, then go for that and integrate those data into your studies as well. Okay, then once you have all these things in mind, then go to the it since it is an interdisciplinary field, then talk to the pharmacologist, bioinformatics. Okay, so that you can create the most comprehensive and the most informative info. You can gather the very informative information from the network you have created. Okay, then envisionate the application of network pharmacology findings in drug discovery. Then always see how you can use this data you have generated for the discovery of a new formulation or the drug discovery, or the if you can find any genes which can help in the plant growth. Okay, so these are the different concepts. Whenever you are designing a project, you should always consider. Then I have told you related to all the important aspect of the network pharmacology, the important aspect and the case studies. So now I would like to conclude this approach. Okay, so the network pharmacology analysis bridges the traditional knowledge with the modern drug discovery. So since we have a lack of evidences in our traditional system of traditional system of medicine, so it helps to bridge the gap between these traditional and the modern drug discovery. So we can use this to create a more solid basis to take these formulation in the clinics. Okay, then through the network pharmacology, traditional medicine formulation are decoded at the molecular level. So same. So it we can facilitate or repurpose the formulation, and we can more understand it more clearly, like how it is being how it is affecting what is the pharmacokinetics and the dynamics of this formulation in our body. Then this integrative approach offers insight into the mode of action of traditional remedies, guiding traditional drug design and formulation studies using cutting edge technologies. Then, by understanding these formulations, we can guide the rational medicinal chemist or the chemist working, the scientist community working in the drug discovery for the better therapeutics and the medications, because there can be certain side effect of the medication as well. We can study those as well with the help of network pharmacology, and we can design the improved or the better formulation. So this was the conclusion of the network pharmacology. Then now I would like to give a brief introduction of our lab. So we are drug chemistry research center, so we are basically based on the Indore. So we are involved in the teaching activities. So teaching activity. So we are providing training in the field of computational drug discovery, okay, and like molecular docking, molecular dynamics, and ligand similarity, as well as the emerging fields like machine learning, data mining, and the deep learning. And we also provide the facility for the collaborative research. So if you have any, you have certain kind of tasks like you want to perform the in vitro activity, okay, or the LCMS, HPLC IL studies. So we can also provide support for those as well. Then we have signed the MOUs with different colleges across the India, majorly from the central region of the India. Okay, so this was about us. So now we are going to conduct several courses in near futures. Like we are providing mini project in machine learning, molecular modeling, MD simulation, and the drug discovery. Okay, we are going to start our program in the molecular diamond simulation using Chromax. So it is a one week program which we will conduct from the 14th of February to the 20th February. Then we are also providing training in the field of machine learning and the talking studies. Okay, so if you want to gain more knowledge related to this field, you can visit our site at dcrc.co.in. Okay, so with this, I would like to thank you all to attend this webinar in the network pharmacology. And if you have any query, you can ask me.
Thank you, Harshita. Uh, can you hear me?
Yes, yes, ma'am,
Hello?
I can. Yes, I can hear you.
Yeah. Okay. So, thank you. You explained it very well. I'm a pharmacologist. I'm from Institute of Chemical Technology in Mumbai, and I'm very much interested in this particular network pharmacology. But then, there is some kind of apprehension when you see all those complex networks. So how does, the analysis, how is it done? I mean, do they give any values when you put, gene target or disease target and compound target, and when you combine them and you get a meaning common, you showed na the thalidomide example.
Yes, yes, ma'am.
So what values do they give? Is it, uh, do you really need to have a bioinformatics person, or even people like us who are not so savvy with the computer can do this? How difficult is it? Or we really need to take some good training for understanding that?
Ma'am, so there are different softwares which can provide a particular number or score for a particular task. So
Okay.
I showed you the Cytoscape. So Cytoscape is a
Yes.
basically software which you can use for the visualization, as well as it integrates multiple modules for the for the topology analysis. Like you can understand how what is the degree of a particular node, means it provides the importance of a particular nodes, means how much nodes it is connecting. And it can you can get the idea related to the hub nodes. Suppose I want to halt two or three processes at a time, and there is a hub node which is involving two tasks. So if I will halt that particular task, or by halting that hub node, I can means I can halt the various processes simultaneously. So you can by using the different tools, you can get all these information. Then there can be other tools like David and the gene enrichment analysis, CAG analysis. By performing this, there is a score for all these tasks. So whenever you
So
will
how do you
study.
come to know about that? Like, because you must have studied and okay, XYZ softwares are there. So where do we get this kind of information? Or do we really need to take a training, proper training in this
Yes,
area?
ma'am. So, ma'am, if you want to wish to take a particular training, it is always better if you can go for the self study. Then there will be the tutorials or the literature evidences. You can go for all these things. Means what is the cutoff people are taking? Like some people take degree as an measure for the selection of a node. some take median for the selection of the node. So it all depends on your need, or how you can how do you want to proceed, what is your reference paper for that particular task. So if you have capability to read all these papers, so you can
Hmm.
go
I feel
go for
that is
it.
important to
Yes, ma'am, literature
understand
survey is
how
very.
people.
Yes, ma'am.
And let me ask you, are you a bioinformatics person?
No, ma'am, I'm basically a chemi informatics person, but
Okay, so
I
basic
do all these.
graduation is pharma.
I'm masters in pharmacoinformatics and PhD.
I thought as well because you said Lipinski and all those things.
Yes, ma'am.
Okay, okay. It is a nice lecture, but I think I really need to learn more. Maybe read papers and understand.
Yes, ma'am. Because whenever you are dealing with a particular value, you have to go through all the literature.
Thank you.
Thank you, ma'am.
Madam, I have a doubt.
Yes, yes, sir.
Actually, partially I know network pharmacology. I have tried. Can you
Sir,
hear me?
sir, I cannot hear you properly.
Uh,
Sir,
partially I know. I
sir,
tell me, madam.
if it is possible, you can open your video as well during the question session.
Madam,
If it,
I have. Can you hear the volume?
I can hear you, sir.
Uh, no, I will put the question and I switch on the video. Actually, partially I know network pharmacology.
Yes, sir.
Okay. When you construct these networks, you get
Yes, sir.
a noodles.
But
Okay.
but
No, no. Let me complete. From that, you said you select a node or a hub, uh, but which one to select because that has to proceed for in vitro. So you have noodles in your hands. So how will you search a particular point? What is the decision? You said some analysis, but nobody
yes.
has discussed on it.
Yes, sir. So there are all these analysis. All these analysis help to find out the various pathways and the nodes. Okay, so you can. I told to ma'am as well. So you can go for the degree based analysis, centrality. Okay, or the most important is the enrichment analysis, because by integrating the enrichment analysis, so you are merging some literature or the experimental evidences with your finding. Okay, so if not possible for that, you can go for the literature evidences. So suppose you have identified 10 nodes. Okay, so one is AKT, EGFR, and some other node. So and you are doing a particular study in the for the cancer, suppose anti-cancer property. So you can take the those node name, and you can perform the you can go for a particular literature survey. Okay, so that you can get your own evidences before going for the biological activity. So relying on the software generated score, as well as your literature survey, it is a very good combination to identify the nodes for the biological experimentation.
No, suppose from our because as like madam, we are teachers, we know much of theory. Okay, so
Yes,
suppose we connect, we get the data. Will you guide us which thing to select? Whether DCRC will help us.
yes, sir. Yes, sir. So you can show us our data. We will keep it confidential, and we will provide you some idea.
That's what because we are able to construct the nodes,
Yes, sir.
but further steps which target to select, we have a confusion.
Okay, sir. So you can share your data. We will definitely provide you support
Yeah.
related to that.
Okay. Okay. Thank you.
Thank you, sir.
Good evening, ma'am.
Yes, sir.
Madam, actually, I am from Ayurveda side. Actually, I am working as a associate professor in Ayurvedic college.
Yes, sir.
Good evening, Manoj sir.
Uh, Good evening. Good evening.
Yeah, I got your reference from Dr. Karthikeyan.
Yes, sir. Yes, sir. Sir, actually, I want to know.
Will you
Actually, I
please
have.
open your video during the questionnaire? Is this possible? Yes, sir. Thank you so
Yes.
much, sir.
Okay. We are working on that. Actually, network pharmacology. The basic problem in Ayurveda, what we feel. I want to share some things. Actually, in Ayurveda, a formulation contains more than 10 to 15 ingredients.
Yes. Yes,
Whenever you
sir.
go for the analysis, actually, this PubChem data and this binding DB, this DigiNet and cake, and after that,
Ye-
actually, I go for the cytoscope also. But the structure actually we have drawn from the cytoscope. What is the complex structure? Actually, I am not analyzing the structure. When
Okay, so
actually the method in which I have analyzed, I have drawn all the things in cytoscope. But after that, actually, I am not recognized what I have did. Again, another thing actually I want to know. Actually, in Ayurvedic formulation, it has been described that it's having some repurposing effect also. Some of the effects of different diseases. Actually, in a one formula, more than 10 to 15 diseases are described in the classical test. So whether we should go for that 15 diseases or whether we concentrate on a specific disease.
so if you want to study the different diseases, you should always go one by one. So just take the data. Your plant data will always remain same. Just change the data of the diseases every time you perform the experimentations. So for one disease and all the formulation, all the plants, okay, then try to identify the targets which are important for that particular disease. Then if it has another indication in another disease, then take those disease target, then map those target with the targets of your plants, so that you can get some ideas.
So you are telling that we should go for only one disease.
One disease,
Then
all plant target, then another one disease on all the particular target. Do not means keep those diseases separately.
okay. Another thing I want to know, our formula contains some the bhasmas. These bhasmas are whether there is some path that actually I can analyze that whether this bhasma is having any target or not. Is there any method?
No sir, actually bhasmas, this that formulation means it is basically what I think about bhasma is it is a traditional nanotechnology kind of thing na. So
Yes.
it's not related to
For
network
example,
pharmacology.
we are preparing iron lava bhasma. Lava bhasma contains ferric oxide or ferric carbonate. Ferrous oxide or ferric carbonate. After the XRD actually we are getting this ferrous oxide. So
Yes sir.
whether there is any rule or anything else by which you can go for this retro pharmacology?
So you can check the targets for the ferric oxide or the ferrous if they are reported in the literature. So because iron is an integral component, it might get target. It might be targeting. I'm not saying it with complete surety, but I just speculating that it can interact with the different biological targets. So you can check the targets of that ferrous as well in our body.
Okay, another thing actually the cytoscope picture, what we get after this KEGG analysis or KEGG analysis, after that actually I go for the cytoscope. But how I can analyze that? Actually, I have drawn that picture.
So
How
you
just I
try
go
to
for the analysis?
so you change the layout of your network first. But whatever
Uh,
you are doing, you go for the pathway analysis. Are you checking for the important module or the pathway analysis, or you are
yes,
just
ma'am, yes.
constructing the? So try
Actually, I
to
am
change the
constructing that
layout.
cytoscope. I have constructed 3 to 4 formulas, but I am not getting all the things. What actually I want to know, how I can explain anyone that what I
So
have
you
done in my
change.
network pharmacology.
Yes, so you change your network layout. Just try to create the sub networks of the common nodes. Okay, so that will be helpful for you to make other people understand.
Okay. Sir, any program by which you can analyze on cytoscope after
So
drawing
there are
in
multiple.
cytoscope?
Yes, sir. There are multiple programs integrated with the cytoscape. So there is an window tools, so which by which you can do all these analysis. You can add different tools in cytoscape as well. You don't need any other software for performing all this. You can integrate all these modules in cytoscape.
Okay, thank you, sir. Thank you, ma'am.
Thank you, sir. Thank you very much.
Thank you so much, sir, for I think there is an echo.
So
You can
one small
Ma'am,
suggestion.
there is an echo.
no echo from my side.
Okay.
Sir, good evening. One small
So
suggestion.
I want to put one more point with the query of Manoj sir, Manoj Kumar Das sir, that yeah, if you are targeting multiple disease in one time with taking the plant materials, so just take one disease one time in one time, and then make a network with plant extract. Okay, sir. And there are many work related to network pharmacology in which many people are using iron bhasma. Okay, so we can do that if you want to collaborate with us. So we are open to collaborate with everyone. And if you have some queries, so you can continue with your query.
Yeah, I would like to add one more thing. Because I'm very humbled that today my teacher, Dr. Rajeshwar Kamalkant, has also joined. So when I was performing my B Pharmacy, he was teaching me pharmaceutics. So I would express my hearty thanks to Dr. Rajeshwar for attending my webinar. So thank you so much, sir. Okay, so
Is there any query from the participants? Professor Prem Kumar, sir.
Sir, I have one suggestion.
Sir, you are not audible.
Okay. The volume is full.
Sir,
Can
you
you
are
hear
still
me?
not audible, sir.
Volume is full.
No, sir, we can't hear you.
Okay. I can hear you.
No,
Hello.
sir.
Okay, I will put in the chat box.
No, sir, you are not audible. Sir, if you have any query, you can write in the chat box. So, Dr. Harshita will reply.
yes, yes, we will try to organize workshop as well in network pharmacology.
Sir, just let me know when you want to this workshop.
Yes.
So we will collaborate and we will organize one workshop on network pharmacology. Yes, it will be online. It will be
Yes.
more better that other people can also join. We will discuss with you, sir, upon the online workshop on network pharmacology.
So thank you everyone.
Yes, sir. Definitely, we will go step by step in the workshop.
So sir,
So, welcome you, Professor Rajesh Kumar sir, Rajeshwar sir. Thank you so much for joining one day webinar on network pharmacology. So, I heard about you from Dr. Harshita. Sir, you are not audible.
yes, sir.
No, sir. You are still not audible.
So you're not
Sir,
audible.
another question, sir, actually, I want to know after network pharmacology, how we go for molecular docking?
So
If you
once
like,
yes, if you identified important targets, you can use those target as an target for the for the molecular docking. So you can dock your phytoconstituent to those particular targets. First, you need to identify targets.
so what is the software by which you can go for this molecular docking?
Autodoc Vina, you can use Pyrex Autodoc Vina. It's freely available and it has good citations as well.
Okay,
Sir,
thank you.
you can check molecular docking via freely available softwares like Pyrex, Autodoc Vina, or Autodoc 4.0, and there are many softwares, freely available softwares. But if you want to do the molecular docking in Schrodinger, so we have good contacts. We can perform molecular docking from Schrodinger as well. And further, we can also proceed for MD simulations from GROMAX as well as Schrodinger.
Okay, thank you.
Yes, sir. Is there any query from the participants? And I want to request Professor Adresa, sir. Sir, you are still not audible.
Welcome, sir. So you need to unmute yourself.
Yeah, yes, you are
Yeah,
being mute.
you are muted. I am facing some problem. No issues, sir.
And one participant, Sajil Sultanat, you raised your hand. Do you have any query?
No, sir, no.
Mr. Sajil, do you have any query? Please unmute
No, sir, no.
yourself on your video. Meanwhile, I want to request all the participants and that we are open to collaborate with you. And if you are willing to collaborate with us for different type of works like some research work, webinars, conference, workshop, so you can contact us anytime. Okay, so I just want to let you know that Dr. Harshita already told you that we are involved in various research fields like molecular docking, MD simulation, network pharmacology, deep mining, data mining, deep learning, artificial intelligence, machine learning, protein interaction, protein drug interaction, drug interaction, and we are also very good collaborations from different part of India, CSIR, IITs, central universities, private universities, and many research labs as well as industries. And we also have good collaboration from other countries, US, UK, South Korea, Australia, Germany, Canada. Many people are working with us. So if you want to collaborate with us, we can do that. And network pharmacology is an emerging field in which you can submit your research proposals as well as you can publish your research work. So if you don't have any query right now, you can mail us or contact us in our number. Okay, so and you can also go through our websites of Drug Chemistry Research Center via login dcrc.co.in. Okay, so thank you so much for joining one day webinar, international webinar on network pharmacology. We are very thankful to Dr. Harshita Diwari for sharing her experience with us and our between our participants.
Thank you so much, sir.